Hello friends, I have already delivered to you two lectures. The first one was on history of microbiology and second one was pillars of microbiology. Of course, this is also part of history. And I may remind you that there is another theme, question, golden age of microbiology. So, golden age of microbiology should be remembered. What is golden age of microbiology? If you see in the dictionary the meaning of history, it is something of past that has gone, time has passed. But if you ask a historian what is antique, he would say something more than 60 years old is the antique. So, golden age of microbiology is also historical. Then what makes this theme or sentence different from history? It is a period starting from 1857 to 1910 when maximum discoveries of microbiology were made. This period, starting from 1857 to 1910, roughly a period of 50 years, has been regarded as the golden age of microbiology. And most of the discoveries were made in this duration. And under pillars of microbiology, under the lecture pillars of microbiology, most of the microbiologists that contributed come in this category. Friend, my today's lecture is on scope of microbiology. What is scope of microbiology? It may also be regarded as future prospects of microbiology or circle of microbiology or work field of microbiology. So today I am going to tell you regarding scope of microbiology. What are the prospects of microbiology? What is the work field of microbiology? And how microbiology can be utilized for the welfare of the society, for the welfare of the humanity. The different aspects, different fields where microbiology can be used, one of them is the agricultural microbiology. So, in the agricultural microbiology, livestock suffers from certain diseases, animals suffer from certain diseases, that is, these diseases are studied under veterinary microbiology, and the person is the veterinary microbiologist. And there may be in the agriculture some root symbionts are very useful, play a wonderful role. Rhizobium is one of them. Many other species, but some actinomycetes, they are symbionts. And by fertilizers, some microorganisms are used as biofertilizers. Some of them are symbiotic and some of them are non-symbiotic also. Azotobacter is a non-symbiotic one, that is a free living bacterium of the soil, and biopesticides. Biopesticides safeguard, they fight against pathogens, pesticides. And for example, a gene from the bacterium Bacillus thuringiensis has been passed to cotton, that is called the BT cotton. The Bacillus thuringiensis is a bacterium, the spores of which, when sprayed on larvae of insects, they are killed because they have got parasporal bodies. That is a protein that is injurious to the gut of the insect, caterpillars, any type of caterpillar. Some of them can kill the mosquito larvae even. So, in agricultural microbiology, biopesticides play a wonderful role. They are useful in the soil microbiology. Soil is the upper strata, upper zone that contains many microorganisms. Of course, it depends from soil to soil. Soil on the bank of the Ganges will be greater number of microorganisms than the soil of the desert or sandy soil. And large number of microorganisms are there. Microorganisms have a great role in formation of soil. Of course, physical conditions, climatic conditions, they also, it is summation of certain conditions, but microorganisms have the major role. And microorganisms may present on the roots, send out certain exudates and attack microorganisms. And microorganisms may be present on the surface of the root of the plants that comprise the rhizoplane. And there may be microorganisms around the, in immediate vicinity of the root, and that is known as the rhizosphere. Soil may be having large type categories of organisms, bacteria, protists, and fungi, viruses, and nematodes. So, not all microorganisms are harmful. Some of the microorganisms are useful. Some of them are used as biopesticides. Some of them can be used as some of them, they have the antagonistic effect. They work against the pathogens. But some of them, for example, fusarium, that causes the wilt of some plants, they are harmful. But, for example, fungi that form mycorrhizal association, they are useful. In orchids, the seeds are microscopic. Millions and millions of seeds may be there in a single orchid pod, but only few of them are fortunate. Mycorrhizal fungus comes as a midwife to rescue the orchid seed in its germination, and later on establish a symbiotic relationship for the whole life. So, soil microorganisms, they play a very wonderful role, have deep interaction with the plants. Then comes the role or scope in aquatic microbiology, that includes the limnological study, marine microbiology, decomposition of the solid and dissolved waste in the water. Limnological study is the study of the fresh water. Many organisms are present in the water. Some of them may be associated. The cyanobacteria, nostoc, anabina, they are nitrogen fixers. Some of them may be symbiotic also. For example, anabina azole is found in a water fern, azolla, that can be used as a biofertilizer. And then marine microbiology, many microorganisms are there. So and so that microorganisms, they have been isolated even from the thermal vents in the ocean. And these microorganisms are could be exploited, and sometimes basic researches are also very useful. Linnaeus once was wandering on the coast of the ocean. He came across a crab that is known as the horseshoe crab. So this horseshoe crab, he named it Limulus polyphemus, and the binomial system of nomenclature he used for it, he did not know at the time that in the coming time this animal will be useful. But the liquid of its circulatory system, fluid, is used in identification of, in detecting the traces of the gram-negative bacteria even. So it is very useful. And one horseshoe crab, in its whole lifetime, gives the fluid roughly about 1 lakh rupees, and we have to import it. But fortunately, in the Sundarban and other oceans parts, some related species have been found, and future is bright that they may be used in place of the horseshoe crab Limulus, and it will save the money of our country. But it is, and many more information may be gained. So microorganisms are of great interest. Horseshoe crab is not the microorganism, but this immunological process, antigen antibody action, it helps in detecting the microorganisms. A basic research turned to be of great applied value, that is used in testing the waters being prepared for the injections in the pharmaceutical companies, pharmaceutical concern, and it is also used in in detecting toxins, bacterial toxins in some other products also. Microorganisms are also of help in the sanitary microbiology, potable water for human consumption, wastewater, sewage, and industrial waste. So potable water is not just that could be placed in the pot. The potable water means that water that is fit for drinking, that is safe for drinking, that may not cause free from pathogens. And then wastewater, and how industrial waste, they could be improved. There are many methods how water could be treated. That includes from placing the wastewater in a pond-like structure called lagoon, to treatment, primary treatment, secondary treatment, tertiary treatment, and that includes even the trickling filters, specialized filters that move on, and a comb-like structure, they drop by drop. So where lagoon is a very simple oxidative pond. So this is the application, a great application of microbiology in the sanitary microbiology. And then in food microbiology, there are certain organisms that spoil the food. and spore formers are particularly. These may be Clostridium, Pseudomonas, Enterobacter, Bacillus, Staphylococcus, fungi, Clostridium botulinum, and of course some probiotics. Probiotics are useful. So, Clostridium botulinum is produces a toxin and causes a disease, that is neurological disease, that is known as the botulism, and is fatal. And but probiotics, they are the microorganisms. Maybe bacteria. They, while taken live, are useful. When they are taken live, are useful. For example, some bacteria that may be present in the curd, that may be present in the yogurt, are useful. Then there is another aspect of microbiology, that is atmospheric microbiology. Here atmospheric, MB stands for microbiology, but the full form, it is better to use the full form microbiology instead of writing this abbreviation. So this application of microbiology in the atmospheric microbiology also. The fungal spores are in the air, bacteria, viruses, yeast, protozoans, pollen grains, diphtheria, and whooping cough are some of the diseases that may be caused by the microorganism present in the air. Some allergens are also there, and the study of these allergens that are present are studied under, and another term is used, that is the aerobiology. Aerobiology. And even our classroom has got so many microorganisms in it. And there is a term, while we sneeze, a fork-like structure is formed, and that carries so many microorganisms, and the fork-like structure, that while we sneeze, some pathogens may be sent outside, and they stand in the atmosphere and may cause sick to the healthy people. And some diseases, for example, diphtheria. Diphtheria is the disease that block the breathing pipe of the children, because the bacteria, diphtheria, that has got the palisade arrangement, one this like that, and in this palisade, they should form a sort of curtain is formed. For example, they are placed one above another, a sort of curtain is formed. So consequently, the child cannot breathe. So many dangerous diseases are there, and even tuberculosis is one of them. That may be because of the certain respiratory diseases are there, that may be very dangerous. Then utility of the microorganisms is also in the dairy microbiology. So the casein, lactose, fat, minerals, vitamins, they are degraded enzymatically. Microbes secrete certain enzymes, and these enzymes, they spoil. They turn, the milk is spoiled, some other products are spoiled, but all organisms are not harmful. Some organisms are useful also. And I remind a narration by the great microbiologist Kluwer. He somewhere wrote about his daughter. My daughter reads in high school. Bacteria comprise a part of her course. She is always afraid of bacteria. Kluwer further adds, But all bacteria are not harmful. Some bacteria are useful also. So, some bacteria and fungi, for example, Penicillium, Streptococcus, Leuconostoc, these are used to provide special flavor to the cheese. Some bacteria are used in curdling the milk, formation of curd, formation of buttermilk, formation of cheese. And the very discovery of cheese was, for example, the camembert cheese was that there was a young boy. He was annoyed because his beloved did not treat him well. He left eating. He left taking milk. So, since he did not consume the fresh milk, he placed that in a very big pen-like cavity in the cave. And in the evening he saw that the milk of sheep and goats he did not take in the morning turned into cheese. Hence came the camembert cheese, famous camembert cheese, and so many varieties of cheese are there that are prepared by treatment with the microorganisms. So, microorganisms are very useful, and some microorganisms of the dairy products are also used as probiotics. The microorganisms also play a wonderful role in industrial microbiology. Drugs are prepared, antibiotics are prepared, steroids are prepared, enzymes, industrial solvents are prepared, and they are of great biotechnological importance also. Their microorganisms are used to treat, find out the solution. They are used in the bioremediation. They are used to rectify the problems created by oil spills. The India-born Anand Mohan Chakravarti, who a US citizen, prepared a bug known as bug bacterium, a species of Pseudomonas, a genetically modified Pseudomonas that has been used in treating the oil spills, remove the effect of the oil spill, break down the oil, and the microorganisms are also used in the formation of single cell proteins that are also abbreviated as SCP, single cell protein. So, microorganisms, for example, yeast has been used as a single cell protein. Spirulina has been used as a single cell protein. Chlorella has also been used as a source of food, and rather chlorella can be taken even in the space. Microorganisms can also be useful in space, that can recycle the waste, and can be recycling of the energy could be through the chlorella, has also been carried in the space by the NASA scientists. And then we have got in our country very promising institutes are there. For example, Central Food Technological Research Institute is there in Mysore, and we have got a very promising recently formed institute, NIFTM, National Food Technology Entrepreneur and Management. NIFTM is a Ministry of Food organ located at near Sonipat, Kundali, in the industrial sector there, and is a very promising institute conducting research on different aspects of food and provides education in different aspects of food. So food industry also has a large number of pickles, organic substances, organic acids, vinegars. They are prepared, and we can also protect our products from spoilage, for example, by refrigeration, bringing down temperature, or by pasteurization, or by some other process. So food microbiology in this way has a very good scope in future. Then microorganisms are of great help in medical microbiology. The diseases like cholera, dysentery, yellow fever, dengue, hookworm, amoebiasis, bubonic plague, typhus fever, leprosy, and malaria, they have been treated. It has been possible to treat them. Bubonic plague has been very dangerous in the past, but now it could be treated. And then AIDS, but AIDS is not treatable even now, and some vaccines are being prepared and efforts are being made. And certain skin infections, they are caused by dermatophytes. Epidermophyton is the microsporum is there. So many fungi are there that causes the or dermatophyte that causes the skin diseases, yet remains a problem. But certain antibiotics have been prepared as nystatin, I told you earlier in some other lecture. And then microorganisms are of great help in treatment of in immunology. Immunological response, smallpox is that has been eradicated, and vaccination for smallpox, vaccination of polio is yet being done. And Russian scientist Metnikov in 1884 discovered phagocytosis, cell-mediated immunity. Cell-mediated immunity, and rather after it, humoral immunity was also discovered. Serological immunity, you may call that while the antigen enters the blood, the humoral immunity, antibodies are formed. So this is of great use, and antigen antibody, they have been used in preparing certain diagnostic kits also, and they have been used in the past also in our country, and certain buildings, certain paints were formed from the urine of the pregnant cow, and something similar to that what we are using the pregnancy test today, that is based on the antibodies and antigen effect. Then there is a very, very bright and recent branch is molecular biology. And molecular biology is a recent facet of microbiology in which we have so many scope. And why microorganism, higher plants are there, animals are there, but why microorganisms only are used in the molecular biology? Because they multiply rapidly, because of their rapid multiplication, and so many scopes are there: antibody formation, serology, hybridomas or fused cells, monoclonal antibody. So these antibody formation, these also have been used in the they are also very used. Hybridoma can be used in preparing some diagnostic kits, and I just told you that for example pregnancy test is there. This is also based on antigen antibody and could be detected just merely by change of color. Why in molecular biology microbes have been used? They are easily culturable, and some of the microorganisms have been used as test organisms. For example, they may be fungi or some bacteria. Escherichia coli is one of them, one of the experimental organism, and Neurospora is a fungus that has been used by the scientists who are studying genetics, and there are some other microorganisms also, yeast, and they have been used in molecular taxonomy also, molecular method based taxonomy. rRNA has been used in the taxonomy. It has been used in finding out the phylogeny, correlating the, for example, in differentiating the archaea and eukarya, that how they are related or how they are different, and some other aspects in identification, in finding out the homology. For example, GC percentage can be used in, or DNA homology, guanine, cytosine percentage can be used in finding the homology. Friends, there is a vast scope of microbiology. We have learned a lot, and more remains to be learned in future, and new researches will emerge, and microbiology has a very bright future. It is hardly 100 years old subject, but has become popular, one of the most popular subject, and most of the microbiotechnological researches, molecular researches are based on the contributions made by the microbiologists. Thank you. My next lecture would be on eukaryotic and prokaryotic cell.